Thank you very much, Daryl. Yeah, really a great pleasure to be here, and really very much disappointed I couldn't make it in the end. Thus is the world around us at the moment, it seems. We were hoping that I was going to be able to make it there, but yeah, that's how life goes, isn't it, at the moment? Fingers crossed we are at the tail end of all of this. So, but still, nonetheless, really glad I can be here, and hope I can return in person another time. So, starting off this first presentation, we've got 3 talks today, and just this first one, we're going to really focus in on soil organic matter and how best to build that using roots, exudates, and microbes. And you know, soil organic matter is an interesting topic. There's been a lot of renewed interest and fresh research in soil organic matter, and I'm going to be sharing a lot of the most kind of current thinking and some of the emerging ideas about how best to build soil organic matter. So this first presentation is the one of the 3 that's going to be a little more dense, a little more technical and heavy. So I put that first off in the morning. So hopefully you've all got your concentration caps on and caffeine in your system. So we're going to go a little bit deeper on this one, and then the next couple of presentations where we will also get a little more applied and a little more practical as well. So they won't be quite so intense, but as Daryl said, I'm going to share lots of scientific papers and research with you, but I am going to break it down and explain it in a nice and digestible way. So this is going to be the theme, the broad topic of soil organic matter, and like always, it's a big topic, like most things are. And as I say, we're going to zoom in and talk a little bit about roots, root exudates, and the role of microbes, and how these 3 components very much interact. And how we can kind of optimize and manage that towards building soil organic matter. Okay, there's my website and Twitter and things if anybody would like some more information on me. Okay, so let's dive right into it, and kicking off. Here's just an outline for the session. We will start by talking about roots and root exudates. Then we're going to talk about how microbes process and degrade and transform those carbon and inputs, and how microbes grow off that carbon, and why that's really important in forming soil organic matter. Then we're going to talk about how we can stabilize soil organic matter. This is a really important piece of the puzzle. People often talk about soil carbon sequestration, but really there is no sequestration without stabilization. We have to stabilize soil organic matter in soils, and that is then the key part to focus on. A few thoughts on nitrogen, a little bit of a link on nitrogen, and that'll flow into some discussions around organic nitrogen. As I'll point out, something that is not discussed enough, that we definitely need to bring discussions about organic forms of nitrogen into the dialogue, into this discussion. And I'll round it off with some nice practical images on the strategies to build soil organic matter. So that's going to be the flow for our talk this morning. Fingers crossed, I will stick to the time. I've done a few sessions recently, and there's a lot to say on this, and a couple of times there I've gone a bit over time, but I've made an effort to be very careful with this one, so fingers crossed. Okay, so let's dive into this. If we were to look historically speaking, you know, over the last 100 or so years, you know, generally there's been this trend, of course, that we've seen increasing yields. You know, we've done well with science and technology, and we've done exceptionally well at increasing yields. Okay, maybe there's been a slight plateau, maybe just in very recent kind of decades, but you know, across the slightly more longer term, there's lots of graphs like this I could have chosen, but you know, here's just 2 examples. Some from Europe, some data from Europe, and some from North America. And as we can see, yes, yields have gone up. Now, if building soil organic matter was as simple as leaving crop residue and incorporating that crop residue and not baling and taking that away, well, you know, we should see a perfect correlation that as yields have gone up, that our soil organic matter has also gone up over the last 100 years, and generally that's not what we have found. Generally, organic matters have been declining. Not always, of course, there's a bit of context there, but generally, I'd say across the board, our soil organic matters certainly haven't been building, they've been declining or maybe somewhat stabilized. And part of the reason, a big part of the reason, is that most of that residue and biomass material that enters the soil, it doesn't stay there. Most of that is processed by the microbes. They degrade it, break it down, and in the process of doing that, they are respiring, they are breathing off CO2. Just like we breathe out CO2, so do the microbes. So as they break down that residue, a really significant proportion is actually lost as CO2, and it's as much as, and more, 75%. I mean, it can be as much as 90% of the residue is ultimately just cycled back out of the soil. So actually you're only left with a very, very small amount of material, biomass, that remains in the soil that can potentially then form soil organic matter. And then it becomes this important characteristic of the organisms, how they're behaving, some of their microbial wastes and residues, some of the plant compounds, how they all interact and intermingle with the soil particles, which finally influences how well it's stabilized. So just to stress that, yeah, most of our residues does go back into the atmosphere, which is why those big yields that we've seen haven't necessarily translated into building soil organic matter. Now, there's a little bit of nuance to this discussion about building yields, and this was from a study in Spain, which was an interesting one. We'll start on the right-hand side here. We can see that right around about the time of the Green Revolution, as we bred smaller varieties, which were higher yielding, they were shorter stature, less above ground biomass, but they put that into grain. This was, of course, a major achievement in terms of feeding the world. So, but what we can see a little bit of nuance to this is that, okay, here we're looking at above ground residues on wheat in this example, and you can see around about this time, okay, yes, yield, yes, things began to increase, but this came at a subtle cost here. You can see that actually the root biomass, the below ground carbon allocation, actually declined in that time. And that's because there's a trade-off. Of course, the more carbon we put above ground to drive yield, the less we put below ground. So we saw this slight decline in root production despite the increase in above ground biomass. And this graph over here on the left is saying the same thing. You can see it's around about the same window of time. We see this decline in the roots, which corresponds with then, yes, okay, increases in yield, yes, increases in above ground residues, and overall increases in primary productivity. So yes, this increase in yield, etc., was really all about above ground, and it came at a cost to the root biomass and below ground carbon allocation, which you can see declining there. So breeding has been a factor that has changed this dynamic, and here's a really nice visual example of this. On the left-hand side we have roots of einkorn, and on the right-hand side we have roots of a modern durum wheat. This is just a simple little pot trial, same soil, same sowing date and growing conditions, just looking at differences in root biomass and root allocation. So you can see clearly here the older variety, the einkorn on the left, clearly is allocating and has allocated so much more carbon below ground to really drive a much bigger root biomass production. So there's a little bit of nuance to this idea that we've been increasing yields. And we could extend this thinking further, and think about the transition from perennial systems into annual systems. And with that change, of course, we've gone to a very annual dominant agricultural landscape these days around the world. And we can see that this has created a shift in carbon down into the soil as well. So the pink, we're looking at annuals, we're looking at crops. And so you can see that, of course, annual crops have a shorter lifespan. So they indeed rapidly allocate carbon below ground. They produce all those root exudates and excrete those and grow those root systems. So they do that very quickly. But of course, as that annual plant moves into vegetative growth and moves into reproductive growth and starts to flower and produce grain or seed or fruit, whatever it might be, they allocate less carbon down to the roots. That drops off rapidly after 50, 60, odd days, max of 100 odd days, of course. this drops right off, because now they prioritize sending carbon and resources into the grain to fill that grain, fill that seed, etc. So we see that annuals have, this transition to annuals has led to a smaller and shorter allocation of carbon down to the roots. If we compare all the green, this is some grassland data for some grassland species. We can see here that they also have a spike early on, but it's then a lot more sustained of a decline. And of course, would be, as perennials, would be influenced by the cycles of the season. You know, there'd be certain jumps in the springtime, and certain it would decline through the rest of the autumn. But overall point is to say that perennials, of course, are going to allocate more carbon below ground to the roots. They know they're going to be here next year. They're going to invest more heavily in those roots, whereas an annual will invest a little in the roots and focus more on producing seed this year. And again, just a nice visual highlighting this. We have, you know, big, deep root systems in our perennial systems, and the transition to annuals has generally led to this change in towards annual plant species who additionally allocate less carbon below. So we've got less roots through this transition. And lastly, another factor here has been fertilization. And it turns out that fertilizers have been very good at increasing yields. They do indeed drive above ground biomass production. But again, there's actually not huge differences across the board within the root biomass allocation. So as you can see here, we're looking at wheat and maize. As we increase the fertilization intensity, moving from the left to the right, you can see that actually, yes, fertilizers do increase above ground biomass, but actually the root biomass is much the same in these two treatments. So just the summary point here is to say that fertilizers are another factor that seem to also drive more above ground biomass and not necessarily improve more rooting and below ground biomass. So there's a trend that we've seen over the past 100 or so years, and that's actually really important, because it is roots and not shoots that actually are the main driver, main contributor to building soil organic matter. It's roots that contribute much more significantly. They play the starring role in building soil organic matter rather than shoots. And there is lots and lots of studies that support this. Here's a bunch of them there, that they all are pointing in this same direction, highlighting the roots being more important than shoots. So, you know, all these factors that have driven yield, that's all well and important for feeding the world, but that has come at a cost of root biomass, and it is roots that are more important than shoots. Now, this, just to give you a rough ballpark of this study here, Jackson, it was a review, so we reviewed lots of other studies that were looking at this, and the kind of the magic figure, or the average figure that they kind of came up with after kind of averaging out all of those various studies, was about this figure of 5 times. So roots make a 5 times greater contribution towards soil organic matter than do the shoots. So just to point out, it's not to say that shoots make zero contribution or no contribution to soil organic matter. They certainly do. They do play a very important role, but it is a minor role. The starring role is all about roots. Shoots are much more important to sit on the surface, to protect that soil. That's really what they do. They create a microclimate. The shoots are there to create microclimate so that underneath that, those roots can be processed by the microbes and efficiently turned into that soil organic matter. So yeah, shoots are still important for erosion, soil protection, moisture conservation, all those other valuable things that shoots do and that we need them for. But it is really roots that we need to think about. So if you want to build more soil organic matter, it's actually really easy. You just got to grow more roots. There's one part of the discussion and part of the answer. So it's all about those roots and not shoots. But what about then root exudates? What role do these carbon compounds play? And root exudates are, of course, these are not the structural compounds of the plant, the root biomass or the shoot, the litter biomass, not the cellulose or the lignin and all of these things, not these structural compounds. Root exudates are part of the metabolic compounds in plants. These are small sugary compounds, carbohydrates, amino acids, even small proteins, and a range of other carbon-based compounds that plants will excrete, will leak or exude out of that root system. And as I'm sure you're all aware, there's been a lot of interest in root exudates in recent years, and with good reason. It seems to be a piece of the puzzle that's also really important that maybe we haven't been focusing on as much, traditionally speaking. So these are highly available sugars, carbohydrates, proteins, all sorts of things in here that are leaking out of the root system that, of course, as you all know, encourage the soil microbes to grow around those root systems. They will feed on those exudates and grow microbial biomass. So what kind of a contribution does root exudates make versus roots or shoots, for example? So let's have a look. A couple of studies have tried to answer this question. And here's one here. We're looking at the evidence for the primacy or the importance of living root inputs, the root exudates this is referring to, and not root or shoot litter in forming soil organic matter. So we have this kind of emerging thought that suggested, well, maybe root exudates are really important for building soil organic matter because those root exudates are highly available, they're highly labile, very easily digestible to the microbes. So they actually could play a very important role as they are excreted into the soil and consumed by the organisms and turned into soil organic matter. So they did an experiment to kind of compare this, and I'll just cut to the chase here, that they show that those living root inputs are around 2 to 13 times more efficient than litter inputs in forming some of these fractions of soil organic matter. So some of the more stable fractions and some of the more cycling fractions. It appears that root exudates are more important than the litter. And so this was from 2018. Here we have another study, now more recent, 2021, also pointing in the same direction. Plant rhizodeposition, a key factor in forming soil organic matter, particularly the stable fractions. So here they found that those rhizodeposits have the highest formation efficiency, 46%, as compared to roots, 9%, or shoots above ground litter at 7%. And here they're talking about, I'm not going to dive deep into this, I'm going to touch on this just in a minute, but here they're talking about a fraction called mineral-associated organic carbon. So this is organic materials that are stuck to soil minerals, mineral surfaces, the clays and the silts, the sands. Of course, sands have much less mineral surfaces. Clay soils have much more, much greater. So clay soils have much greater potential to form this mineral-associated organic matter. It's a mineral-organic carbon kind of complex. And when we get this binding, it's forms a very stable fraction of soil organic matter. So anyway, the general point there is to highlight that there's quite a contrast. Root exudates are very effective, highly efficient at forming this very stable fraction as compared to the litter inputs. So here we have ultimately two studies that are pointing in this direction that really highlights that, well, actually, root exudates are very, very important. And indeed, they may be more important. Certainly, they have a higher efficiency in which they are converted into soil organic matter. And so that becomes then really important is, well, how do we optimize root exudates? How do we optimize plant growth, plant health, plant production, as well as root exudates, in order to make sure that we have lots of these compounds leaking out of the root systems to feed microbes, which then those carbon compounds are very highly efficiently converted into stable soil organic matter. So however we can manage photosynthesis and optimize plant growth through the growing season becomes really, really important. So, and things like optimizing plant nutrition, not that we're not going to talk about this topic today, but optimizing plant nutrition, optimizing the availability of all the macro and micro minerals, the essential minerals, towards plant growth. Those minerals are the catalysts of photosynthesis and plant metabolism. So one of the really important ways to optimize root exudates as well as plant production is through optimizing plant nutrition. So let's have a look at some of the other factors that influence the quality or the quantity of root exudates. And the species, the different plant species is a really important factor. So grasses and different grasses or different cereals, or versus legumes, versus brassicas, versus other broadleaves, all the different kind of plant species, they all release rather unique and different root exudate cocktails that are unique to that species. And even it goes down to a varietal level. There are some varieties that have different root exudates than other varieties. It can be down to that level of specificity. But stepping back from that, yes, okay, we know the brassicas release a very different set of root exudates than new legumes, than does a cereal, etc., etc. So plant species is a really important factor, and this is where if we integrate some level of plant species diversity, maybe some intercropping or companion cropping or cocktail cover crops, for example, then we're getting a greater diversity of these exudates coming out into the soil, which can have some benefits for the soil microbiome. The plant age, the plant stage, developmental stage is an important one as well. We saw this on the graph earlier, where annual plants, for example, they will release most of their root exudates in those early vegetative stages, but then exudates drop off rapidly as the plant moves into the reproductive stages. So keeping plants in vegetative stage of growth keeps root exudates turned on, and that's why plant and livestock integrated systems, be that grazing and cover crops or grassland pasture systems, this is why they're very good at building soil organic matter. That grazing activity of the animal is a disturbance which pushes the plant back into vegetative stages. That keeps the root exudates turned on as it develops, and then of course it's grazed again and knocking it back. So vegetative plants are really important for maximizing root exudates. Plant nutrition, as I just touched on before, their role of minerals, of magnesium and nitrogen, they form part of chlorophyll and helping to kind of be the side of photosynthesis where we can bring carbon dioxide in and build sugars and carbohydrates and build all of those root exudates that get leaked out of the soil. So plant nutrition is a really important one. And then, a bunch of other factors from there. Neighboring plants. This is an interesting one where we now know that plants also can sense each other. They communicate through gases, through smells and scents, volatile organic compounds, where they will release these volatile organic compounds and detect each other. And that's been shown that can also change root exudation patterns, detecting neighboring plants. So again, that's an opportunity within the companion cropping and intercropping space to optimize companions that actually are very synergistic through some of that interaction. And then we have, other interacting organisms. Microbes growing around roots can change the exudates. Insect herbivores, a pest attack or disease attack can change root exudates. The grazing animal herbivores can also do the same. So lots of kind of factors there. And then a range of other environmental factors. So things like, standard things that might influence photosynthesis, light, temperature, soil nutrient availability, soil moisture. All those kinds of things that influence plant growth ultimately will also influence root exudates. So there's just some factors maybe there to think about in terms of what management can we do to optimize some of these things in order to improve root exudation coming into the soil. So, okay, what comes next? We have roots entering the soil, we have those root exudates entering the soil, and now we shift attention onto the role of the microbes. It's the microbes that have to process those carbon inputs, be that litter, be that exudates. They have to process it, they have to break it down, metabolize it, and then ultimately turn this into soil organic matter. So this is the shift now into the microbial world. And what we see is that, okay, plants are still really important. Microbes are key in transforming and breaking down those carbon inputs, but it's still up to plants, it's still up to photosynthesis, which is where the carbon is coming from, to then integrate that into the soil. And so when that plant material enters the soil, be that roots, shoots, or exudates, of course the microbes will respond to that. They will feed. There's a readily available food source. They will begin to feed and begin to grow their microbial biomass, multiply as they feed and feed and feed, and they go through this process of growing and dying, growing and dying, growing and dying, kind of continually using the available resources. So they will respond to that carbon input, they will grow, they will feed, and as they kind of use up the resource, they begin to die once they stop feeding. And as microbes begin to die and decay, as they then begin to break down and decay, their cell walls and their bodies and their, this is called microbial necromass, their dead bodies, not biomass, but necromass, and then these various microbial waste products and microbial residues, microbial detritus, all of this kind of material can then get stabilized as soil organic matter. This is particularly relevant for, again, medium or clay soils that have a lot of these mineral surfaces, because we get this kind of interaction between mineral surfaces and the dead microbial bodies and their cell wall components. We get this chemical interaction between mineral surfaces and these dead microbial components, where they form this very stable mineral associated organic matter. So that's kind of the process. It's all about growing biomass and then stabilizing the dead biomass or the necromass as soil organic matter. And we have a whole new school of thought in the world of soil carbon science, realizing that actually that this microbial necromass, these microbial residues, they actually form a very significant fraction of soil organic matter, much more than we previously thought. So here we have one of the first studies that really tried to answer this question: what is the actual quantitative amount of the microbial necromass? And here we show that microbial necromass can make up more than half of soil organic carbon. Hence, we suggest that next generation field management requires promoting microbial biomass formation and necromass preservation in order to maintain healthy soils, ecosystems, and climate. Okay, so it's this 50-odd% of what we have always thought about soil organic matter. Turns out that 50-odd% of that is these dead microbial bodies and their waste products, this necromass and their waste products. So they're very important. They make a very important contribution. And of course, okay, that's if that's around about this 50-odd%. Okay, there's still the other 50-odd% that is made up of plant materials. So we have this kind of merging thinking, highlighting that, okay, we have two distinct kind of fractions of soil organic matter. Some is still plant remains that are in various states of decay, plant materials that have been broken down by microbial enzymes that kind of break them down. And then we have this fraction of carbon material. Of course, again, it was once plant material. It all still comes from plants anyway, but it got ingested by the microbes. They consumed it. They turned it into microbial biomass. They turned it into necromass. And so it turns out that generally we see that the plant residues, the plant material, takes a leading role in forest ecosystems. This is where we see more plant-related soil organic matter. Whereas in grasslands, we see that the microbial contribution is much greater. So microbial biomass and necromass is really important in grasslands. The plant litter is really important in forests. And it seems that agricultural lands are somewhat in the middle. Your croplands is somewhat kind of in the middle, this equal kind of contribution of plant and microbial remains. So let me just read a few quotes from this paper. As it says, microbial necromass on the rise, the growing understanding of its role in soil organic matter development. I'll just read a few quotes here. It had long been believed that remnants of decayed plant matter were the main components of persistent or stable carbon in soils. Increasing evidence has led to a paradigm shift. Dead microbial mass is the dominant component of particularly the long-lasting or more stable fractions of soil organic carbon rather than decayed plant material. Accordingly, this evidence is shifting research, focusing from the humic matter to the microbial contribution. However, of course, there are plenty, like always, there are plenty of knowledge gaps. There are various interactions here that we have yet to uncover. So there is still some knowledge gaps. But what we are now seeing this clear picture that, okay, the processing of the carbon by microbes, the transformation by microbes is key, particularly then how they die and stabilize. And so here's a nice image that kind of just compares these two schools of thought. Here we're kind of looking at, as I say, the old paradigm of decay, of plant material contribution versus the new paradigm of the microbial contribution and how this works. So what we're looking at here is, picture this being some plant material, some plant residue, something like lignin, something very complex. The microbes, they can't just feed on lignin. As you all know, your crop residue, your straw, etc., it has a very wide carbon to nitrogen ratio. It's full of complex structural compounds, again, like lignin. The microbes can't just digest that. They can't just absorb that big complex molecule. They have to break it down into smaller constituents, which then their bodies can absorb, which then can help them grow that microbial biomass. So the microbes, they produce these specialized enzymes. They will release these enzymes, exude them out of their bodies. And these enzymes are like scissors that then take that complex molecule and begin to cut it up and break it down. So they are producing these are called extracellular enzymes, these specialized microbial metabolites that cut it up and break it down. And when we get it broken down into these smaller constituents, well, then these small pieces is what then the microbial cells can absorb, and that's what they will ingest. So you can see this was the previous school of thought that kind of thought about, okay, it's all about breakdown, it's all about decomposition. This is what catabolism refers to. It's all about breaking down plant matter. And on one hand, the school of thought kind of said, well, actually, we want to slow decay. We want to keep plant material in the soil. We want to keep this material in the soil for as long as possible. We want to increase the residence time. The longer it lives in the soil, the more benefit it can bring. And on one hand, we were therefore suggesting maybe we want to slow this decay and keep more of these complex materials in the soil. But it turns out that that's, again, not the full picture, and maybe not therefore the best way to optimize things. We want to be working in both spheres. The reality is that once these materials are broken down, okay, then they are absorbed by the microbes. So then it takes in all those small pieces and it ingests it. But then what does the microbe do? Then it grows its body. It grows its microbial biomass. So again, it uses other enzymes, internal enzymes like sewing needles, that help it to resynthesize or to rebuild. This is called anabolic processes, where we then take those smaller pieces and we stitch them back together and build microbial bodies, build microbial cell walls, build microbial structures, et cetera, et cetera. We build microbial biomass. So this is what they're saying, is it turns out that actually what may be more important is how well the microbes are rebuilding their bodies, not necessarily how things are breaking down, but how well or how important we are building, resynthesizing, growing microbial biomass perhaps is a more important focus, as we now know that 50% odd of soil organic carbon is made up of these resynthesis or microbial components. So that's the subtlety of the shift, and that subtlety is really important when we then think about the two food sources that I touched on earlier that are really important for building soil organic matter. We talked about crop residues, be that roots or shoots, and we talked about root exudates. And so if we acknowledge that there's a very important microbial contribution, that dead microbial bodies form this 50-odd% of soil organic matter as well, then the obvious question becomes, well, what is the best way or the most efficient way to grow microbial biomass, to help them grow more microbial biomass, which will then form that necromass, which will then be stabilized as soil organic matter? And so if we then ask that question, we have this concept of carbon use efficiency. And for all of you ranchers and cow guys tuning in the room, of course, you'll understand this in terms of different food stocks for livestock, some which are more easily digestible than others. So, of course, if a microbe is looking at these two food sources, this one over here with the wide carbon to nitrogen ratio, lots of lignin, lots of complex compounds, of course, those microbes have to expend a lot of energy to break that down. They have to release all those scissors, those enzymes, and cut that up. And that's a drain on energy. They have to produce all of those things to break that litter down, that complex material down into those small components, which then they can ingest, which then they can absorb. So that's a lot of work. That's a lot of metabolic work for them, energetic work, versus these root exudates, which are highly labile, highly available, highly digestible. And literally this concoction of smaller, simpler, low molecular weight compounds, not these complex stuff, but small, simple stuff, the metabolic compounds, sugars, carbohydrates, amino acids. These, of course, can be immediately and instantly ingested by the microbes. Highly digestible, highly metabolized into their microbial biomass. So therefore, this is this nuance about efficiency. Root exudates have a much higher carbon use efficiency. They are much more efficiently converted into microbial biomass than is stubbles or root litters. And this picture is just really explaining that same concept. here's to say that, okay, the microbe cannot ingest this complex lignin, this complex material. So it has to, here's the key point, it has to expend all that energy to release, using up its metabolic energy to release these enzymes, these scissors that cut it all up, and then it will ingest it, and then it'll grow, build its body, build its cell walls, etc. And again, as I mentioned, those cell wall constituents, as they die and decay and break down, they can get stuck to the soil particles or wrapped up within aggregates and stabilized. So the key point here is to say that, well, if we were delivering just these food sources like root exudates, well, of course, the microbe doesn't have to expend all that energy to produce these scissors to cut this up. If we just give it some highly available food, highly digestible, it will rapidly absorb that, save all that energy on producing those scissors, and put that energy into growing more microbial biomass. So per unit of carbon, we will grow more microbial biomass. And that's what carbon use efficiency is all about, and the root exudates having that higher efficiency that we talked about in those couple of studies. Okay, so that's the really deep kind of dive into that picture, and this is why we have this emerging interest in the role or the contribution of root exudates. It's that highly efficient nature. And again, in the same way that I said, it's not to say that just because roots are more important than shoots, that we downplay the value or the importance or the contribution of shoots. They still play a piece of the puzzle. In the same way that root exudates are not necessarily the be-all and end-all either, as compared to roots. You know, they all play different and distinct functions, and the message is just to say that all three of those should be acknowledged, and maybe they need to be managed differently, accordingly, because they're all different, that they're not just one thing. There's nuance there, they are different, and therefore the controls and how we manage them maybe should also be different as well. Okay, so that's the key point. Now, just to then kind of wrap this up, I'll try not to go too deep into this. We could talk about this for a long time, but I just want to kind of summarize then the key points here. So when carbon comes into the soil, when those plant residues, the litter, etc., when it comes into the soil, we would generally call this particulate organic matter. This is that plant material that is in various states of decay. It's in a continuum of decay. This is the plant material that is being attacked by those scissors. It hasn't yet entered a microbial body, is the key point. It's still plant material. It's in various states of decay. It's been attacked by those scissors, but it has not yet been absorbed by a microbe. This is the key point. So this is what's kind of called particulate organic matter. It's still working its way into the system. It's still being broken down. It's still it's active and it's being cycled, but it's not yet been absorbed by the microbe. And this is a fraction of soil organic matter, often termed particulate organic matter. And the key point is once that has broken down enough or to become small enough where it can then be absorbed by the microbe, this is then where we have a shift. Now that particulate organic matter is now formed microbial biomass. The carbon has entered the living organism. And again, some of that carbon can be lost as breathing, and we talked about that at the beginning. Significant amounts of carbon are lost through this process, the CO2, through the breathing of the microbe. But key point here is now that carbon is inside a microbial body. It became available, and therefore it is now inside a microbial body. Now that microbe, of course, can grow and die, grow biomass, produce waste products, excrete things. And when that microbe dies, it is also then goes through states of decay where we now have some bits of kind of organic matter, soluble or dissolved organic kind of materials in the soil solution, which then can stick to the soil particles, this mineral associated fraction that I talked about. So we have some highly available breakdown products and cell walls and these kinds of things that then can get stuck to the mineral surfaces. And this becomes then highly stable soil organic matter. Now, it's not to say that it's permanent. However, it can also, some of that organic materials can, as highlighted here, desorb or come off the mineral surfaces back into the soil solution where, okay, it can also be reused and recycled. Some of the current microbial biomass might then feed on that material. And then again, they're going to grow and die. There's a cycle happening here. There's a cycle happening here. Okay, so just what I really want to point out is that, yes, this is a very stable fraction of soil organic matter. It's called mineral associated versus the raw plant material in a continuum of decay. That's called particulate organic matter. And the key point is how efficient or how well we can get this residue material into the microbial body. And once we've got it into the microbial body, once it's digestible enough, like those root exudates, once we've got it into the microbial body, it can go through these various interactions, which can then stabilize it in some of the more stable fractions. And as I mentioned, this is key. It's not just about your carbon coming into the soil. It's not about biomass coming in. It's not about microbial processing, although that's really, all those things are really important. It's about how we stabilize carbon. There is no carbon sequestration without stabilization. And so there are two main mechanisms by which that material can be stabilized, be that if it's in particulate organic matter or be that if it's this mineral associated stuff. We can either chemically or physically stabilize and bind that that organic material into as soil organic matter. So as I've kind of touched on, alluded to already, the chemical stabilization, this is referring to these various organic fractions, organic molecules, and again, these could be plant materials or it could be microbial materials, the microbial bodies that we touched on. It's when these carbon-based compounds stick to the clays or the silts or a little bit on the sands, sticking to the mineral components of the soil. This is a chemical attraction. This is called an organo-mineral bond, and it is a chemical process in which the carbon is then chemically attracted to and stuck to and stabilized to the clay surfaces, to the soil colloids, etc. So that's one way in which soil organic matter can be stabilized, and that's, as I mentioned, called that mineral associated organic carbon. And the other way is a physical binding, and this is all about aggregates. Okay, and I'm sure you'd all be familiar with the importance of aggregates, those microbial glues that help to stick soil particles together. You know, earthworms and insects that then burrow through and also push particles together, forming these aggregates. And when we form aggregates and soil particles all stuck together, of course, we create that very valuable pore space in between. And it's that pore space in between the aggregates that, of course, is really important for your infiltration. When we do the infiltration tests, etc., we have good pore space, good pore connectivity, which lets that moisture infiltrate from the surface and percolate down through the soil. That pore space is only formed because we have formed aggregates sticking soil particles together, creating that pore space. And that physical pushing and sticking together to form these little balls or these aggregates, that is another mechanism by which we physically can trap soil organic carbon within those aggregates. So this is a physical process of just wrapping it up, sticking it in a ball, where it is then inaccessible to the microbes. They can't access it, they can't reach it. So we kind of have a physical stabilization process. And here's an example here. You know, a root was growing past this aggregate. It sent a little root hair into the aggregate to feed and get some nutrition out of the aggregate, for example. But over time, as roots grow and die, as you see, as the root dies off, we're left with that little kind of root hair still embedded within that aggregate, where it is, again, protected from microbial attack, protected from those microbial enzymes. So therefore, we have physically stabilized the soil organic matter. Okay, so that's your two mechanisms, and that's really important. Okay, so moving on, let's then talk a little bit about some of the nutrition, and we'll wrap it up with some practical examples. So I gave this slide already. We talked about the role of fertilizers and how we increase fertilization. This has led to greater above ground biomass, but not necessarily more below ground biomass. And we have other studies that support this. Here we're looking at, as we increase nitrogen fertilization, we're looking at the amount of carbon allocated below ground. And we see this graph is actually a summary of 3 other studies. And as you can see, that we see this general trend that plants will allocate less carbon below ground, less to the roots, as we increase nitrogen fertilization. So there's a nice example of highlighting this effect of fertilizers enhancing above ground, but maybe at a cost to below ground. And then here's another study that I think really encapsulates the essence of this discussion about how fertilizers, how nitrogen particularly interacts with carbon. I think this slide kind of really answers the question really nicely. the summary point is to say that both insufficient and excessive nitrogen fertilizer ultimately can lead to reduced root biomass production. So there's a happy medium. So you're looking over here at these graphs. These are just from 2 different seasons of the trial. One was a bit wetter, one was a bit drier. We see a similar pattern, a similar trend. So here's our yield in green. The green dots is yield. And so you can see that as we increase nitrogen across the bottom, as we increase nitrogen, yes, we are increasing yield up to a certain plateau. And you can see it. There it is there. It plateaus off. Same thing happens the following year. So yes, nitrogen increases yield. However, where it got really interesting was this red dotted line here. This is the root biomass. And so you see here, yes, low nitrogen induced lower root biomass because the nitrogen was deficient. If the plants are deficient in nitrogen, then of course they haven't got enough resources to grow that root biomass. So we start off with less root biomass, but then it increases as we increase nitrogen to the peak. Okay, and then as we increase nitrogen, root biomass begins to decline. And we see the same thing here. So the summary point, okay, I've just got a quote here from this. Root mass was maximized at 168 kgs of N in this example. Zero and excessive fertilization decreased the root mass by 33 and 17%, respectively. Okay, so that sums it up perfectly. If you're low on nitrogen, you're going to have a negative impact on root biomass. If you've overshot nitrogen, you're also going to have a negative impact on root biomass. So this is how and why excess nitrogen can have a negative effect on soil organic matter. It's because too much nitrogen induces less root biomass production. And this, as I say, this study here summarized that, and this one brought that context of both low and high. So the summary point here is just to say that, yes, it's really important to optimize your nitrogen. You don't want to go too low. You don't want to be too high. We have the mechanisms, the understanding of how and why that excess nitrogen can indeed be a big problem. Now, excess nitrogen can also have negative effects on the soil microbes, on the microbial biomass. And as we've just talked about, microbial biomass turns into microbial necromass, and that's really important for building stable soil organic matter. So it turns out here that here we're looking at a meta-analysis. So this is a study that looks at lots of other studies and kind of reviews them all and reanalyzes the data and kind of tries to average them out or look at the overall overarching kind of trends or patterns. And here they're finding that that indeed nitrogen can have some negative effects on microbial biomass. So I'll just read a few quick quotes here. We conducted a meta-analysis, 1,248 data points from 120 studies, looking at 120 different sites that cover many different soil types and climatic conditions around the world. And the results showed that nitrogen enrichment significantly decreased microbial biomass carbon and arbuscular mycorrhizal fungi across all studies. Our findings highlight that nitrogen enrichment decreased microbial biomass and showed a significant effect on some of those microbial enzymes, those scissors that I talked about earlier, across a range of these different ecosystems. So therefore, you can see here that potentially we still need to be careful with nitrogen also, because it can reduce microbial biomass carbon, it can reduce mycorrhizal fungi, and again, those components turn into those microorganisms die, turn into necromass, and form that very stable soil organic matter. So there are good reasons to be very cautious with our nitrogen inputs and optimize. And I know that's easier said than done, but trying to optimize the nitrogen inputs and align that to crop performance, that's going to help to minimize some of these negative effects on root biomass production, but also some of these negative effects on the microbes as well. But what form of N? You know, a lot of these studies that I'm talking about, of course, they're always looking at synthetic nitrogen because, okay, that's the form of nitrogen that we mostly use in agriculture. So they're trying to, which is fine, that's practical reality. They're trying to see what are the negative effects or what are the potential effects of the way in which we do things at the moment. But if we were to open that discussion and say, well, okay, does synthetic nitrogen behave differently to organic fractions of nitrogen? What influence do they have on roots or on microbes? And it turns out that that's a really important nuance that is often left off in some of that earlier discussion. So let's have a look and break apart this now Pandora's box. So what are we talking about? We're talking about inorganic versus organic nutrient sources, okay, nitrogen in this example. An inorganic nutrient is just the nutrient itself, okay? Nitrogen could be nitrate, could be ammonium, for example. An organic form of nitrogen is that same nitrogen atom, but it's embedded within some carbon. And that could be an amino acid, that could be a protein, that could be all sorts of compounds. So really, an organic nitrogen really just means a combination of some nitrogen and some carbon, which forms some kind of an organic mineral. So now we have organic nitrogen. It's nitrogen wrapped up in carbon or embedded within carbon, amino acids being a good example. So the point here is to say that, well, indeed, nitrogen on its own behaves very differently than nitrogen when it is embedded in carbon. That carbon changes the behavior of that nitrogen atom. Microbes love carbon. They like to feed on this, and this carbon has lots of charged surfaces. It can bind to other charged surfaces. It's more stable in the soil. It's a little less reactive. So there are some subtle differences between these. So when I talk about soil organic nitrogen, what are we talking about here? Actually, most of the nitrogen in soils is in the organic fractions across the board. Of course, immediately after you apply some artificial fertilizer, of course, there's a spike at that time. So that changes the dynamic there. But actually, a lot of the nitrogen in your soils is already in these organic forms, embedded within soil organic matter, actually. So that's things like proteins and peptides, which are kind of small proteins, amino acids, amino sugars. These are components of those cell walls that I talked about, microbial cell walls that get stuck to the soil surfaces. So these are some of the types of things we're talking about when we are referring to what is soil organic nitrogen. These are the types of things. Most of that organic nitrogen is embedded within soil organic matter. And okay, the classic view, therefore, of nitrogen nutrition and nitrogen management, or from a plant nutrition point of view, we've always said, okay, well, we want to cycle our organic matter. We want to take some of those, okay, the proteins, for example, within the soil organic matter, and we want to break it down to some smaller constituents, like those peptides. We want to break that down again into amino acids, and then we want to mineralize the soil organic matter. We want to mineralize that to release the inorganic fractions, like ammonium, which can then get rapidly converted into nitrates. And this has been the kind of classic view of plant nutrition. It focused heavily on the role or the contribution of ammonium and nitrate, those inorganic fractions. We've always talked about organic matter needs to be broken down, needs to be mineralized in order to release the nutrient where it can then be taken up. So classic plant nutrition has viewed heavily on these inorganic fractions, but it turns out that plants can indeed absorb and use these amino acids and peptides and proteins. Plants can use these more complex fractions of soil organic matter. So this graph here is summarizing this really nicely. It's not just about inorganic fractions. Yes, they form a piece of the puzzle, and yes, our understanding of those and their role in plant nutrition is definitely the greatest. But as we move this way back towards these more complex compounds, substrate complexity as it increases, indeed our understanding of these materials in plant nutrition becomes less and less. But the key point is just to say that we know that plant roots, some root exudates, they are these proteases. These are the enzymes that cut apart that protein, cut apart that organic matter and break it down. So it turns out that yes, plants can absorb amino acids directly and even larger proteins directly. They can absorb these bigger molecules, not just the smaller inorganic nitrogen compounds. And as we also now know, plants can even engulf and digest whole microorganisms directly themselves. So I'm sure some of you have heard of this rhizophagy cycle. So obviously, there was some debate. Many people have said that, well, plants can't take up proteins. Proteins are too big, they're too complex. But as we now know, plants can take up whole microbes. So a whole microbe is a lot bigger than a protein. So if plants can take up bacteria, they can certainly take up these more complex proteins. So we don't have to feed just inorganic fractions of nitrogen to plants. Some of these other organic fractions, aminos and proteins, are very valuable, make a very valuable contribution to plant nutrition. However, there's a lot of knowledge gaps on this. Our understanding of the roles of this is certainly limited, but that's beginning to change. We're now recognizing the value of these organic nutrients. And I actually just put this slide up here just more so to say something else, but normally this is about kind of nuances between how ammonium and nitrate are processed in the plant. Key point being that when ammonium is taken up, it's metabolized in the roots. It's converted into amino acids in the roots. Therefore, generally, it encourages more root biomass. Whereas nitrogen is taken up by the roots and then sent up into the leaf where it is processed. And therefore, we find that nitrates generally encourage more above ground biomass, more shoot biomass, because that's where the nitrogen is converted, versus ammonium, which is converted in the roots. Therefore, it encourages more roots. And that's what this slide is about. But I just wanted to add to that and actually just highlight and say that that organic sources of nitrogen, when the plants absorb these amino acids or proteins that we were talking about here, it's the same pattern. Those forms of nitrogen generally encourage more root biomass. So the organic fractions of nitrogen encourage greater rooting biomass, rooting production. Those aminos and proteins, they generally stay in the roots and therefore encourage more root biomass. That's the key message. So organic fractions of nitrogen, these aminos and proteins, etc., they generally encourage more rooting traits, of a rooting phenotype in the plant. Okay, so that's really important because, as we've also just discussed, it's roots that are more important for building soil organic matter. So actually, by shifting nitrogen management towards more of these organic fractions, actually that can be also a mechanism by which we can encourage more rooting, more root biomass. And it turns out that then that's also really important for this discussion about, well, how does nitrogen interact with soil organic matter, and does nitrogen build or burn soil organic matter? And there's that little bit of nuance there, as I touched on earlier. Yes, I support the idea that there's an optimum. Too little nitrogen will cause not enough root biomass. Too much nitrogen will cause not enough root biomass. Optimum nitrogen will give us that perfect curve. But of course, that's talking about inorganic nitrogen inputs. If we think about this conversation in relation to some of those organic forms of nitrogen, well, those organic forms of nitrogen, which is nitrogen embedded with carbon, again, as I highlighted, they seem to encourage more root biomass, more root growth. And these are fractions of nitrogen that, again, do not contribute to some of those declines of microbial biomass that review study that we talked about highlighted. So what I'm trying to say is that these organic fractions of nitrogens, they're very favorable towards microbes. They're very soft, very easy on them. They prefer these organic fractions. It's kind of good for them to feed on and grow. So organic forms of nitrogen shifts this scale in this direction, where we encourage more roots. We have greater association between microbes. And it is this then that tilts the scales towards this process of organic matter stabilization, of building soil organic matter, when we have more organic fractions of nitrogen cycling in the system. Conversely, when we have more inorganic nitrogen in the system, well, as I mentioned, that encourages more shoot growth, more above ground growth. Inorganic fractions are more leaky. There's greater leaching, greater losses from inorganic fractions than organic fractions. So it is a dominance of inorganic nitrogen in the system that then can tip the scales a little bit in that direction, which can lead to more of the burning of soil organic matter or the soil organic matter decomposition. So I think this is just a really nice summary slide, and it's just to highlight that both are a factor, both play a role, and it's not that we want necessarily one or the other. We kind of need a combination or a balance of both, and it's just a question of which way are the scales tipped. And so the summary point is just to say that the more organic fractions of nitrogen that you can use and integrate, which is what we'll now go and talk about how to do that with our remaining time, the more you can steer things in that direction, the more you're going to be steering things towards soil organic matter building, is the key take-home message. So what are some of the sources of organic nitrogen that we can use? Of course, it's going to be any crop residues. You're going to have some nitrogen in there. Of course, legume residues are going to have more than cereal or residues, for example. So crop residues, particularly roots, are going to be very important. Some of those root exudates are also contain nitrogen. For example, some of those amino acids the plants are also releasing as root exudates. Some of the microbial byproducts, the microbial waste products that we talked about, some of those will have organic fractions of nitrogen. The dead organisms themselves, so the microbes or other soil insects and their waste products, you'll have some organic nitrogen there. And then, like from a practical point of view, organic amendments, so things like composts or manures. Of course, anytime you're applying compost or manure, you're going to have some organic nitrogen fractions entering the system as well. So therefore, in terms of practically speaking, getting more organic nitrogen into the system, as much as we can all appreciate the value of organic amendments of compost and things, I know that's not practically feasible for a lot of broadacre scenarios. So therefore, really one of the easiest and kind of universal strategies to get more organic nitrogen into the system is with legumes. And I think, therefore, the idea of companion cropping or intercropping is a really good one. I'm glad to see this trend gaining traction at the moment. I think having then a combination of legume and, oilseed, we've got some canola here in this example, and peas, or legume and cereal, it could be whatever. Having these together gives us a really nice balance of roots and root exudates and residues. Of course, I'm sure many of you maybe don't like growing pulses in your rotation because the pulse residue breaks down too quickly and doesn't give you soil protection. And of course, that's really important in a dry land farming system. And that's why I think I'm also not a big fan of monocultural legumes for that reason. However, if we have legumes, again, with a cereal or an oilseed, well, then we have the perfect combination in my mind, where we have the legumes providing that organic nitrogen, very friendly for the microbes, great, they're highly digestible. Therefore, again, that point about being highly digestible means that it's easier for the microbes to break it down and therefore form that microbial biomass and necromass that we talked about. So, of course, legumes, they have a lower carbon to nitrogen ratio than cereals. So legumes break down quicker, more easier, as you all know visually, but that means that quick breakdown means that it's more, with a higher efficiency, means that it's more likely to form more stable fractions of soil organic matter. So I think it's the best of both worlds. You're getting some really good quality, high quality litter in the legumes being integrated, but you're left with, some oilseed stubble, or even better, some cereal stubble to provide that soil protection that you're all looking for. So I think the companion opportunity is a great one and the easiest way to bring more organic nitrogen into the rotation potentially every single year. Just another example here, just to visually illustrate this, the idea of kind of undersowing a legume companion, for example, I think is also a great idea. And I know maybe in some dry, this is from the UK, and where they'll have a bit more moisture, that's why that clover looks green and happy underneath. I know in a drier environment that may not always be quite so possible, but visually it's just a really nice example here of, we go through with the combine, we're left with living roots, we've still got living roots, we've still got root exudates. As we've talked about, those root exudates are really important for feeding the soil biology. And again, we've got this nice balance of stubble, of high carbon to nitrogen ratio material for soil protection, as well as some better quality litters for soil feeding. So again, this idea of kind of maybe an understory companion, a clover, for example, I think is also a really great idea. Okay, so let's wrap it up with just a few, I'm going to wrap this up with just a few photos and images of, therefore, some tips to kind of try and all of this kind of detailed information we've gone through this morning. Let's put it into some practical terms of how we can, at a field level, implement some strategies to build soil organic matter. Number one, well, obviously, as many perennials that you can integrate into your system, the better, or at least a perennial phase within the rotation. I think there's a lot of opportunities for that. Maybe within cropping rotational systems, having a 3-year perennial phase. Great if you could also have livestock, of course, livestock graze that so you can maintain an economic income stream. But, have a 2 or 3 year perennial phase. Have that livestock interaction, grow lots of roots, below ground carbon. As I think if you can get more perennials into the system across the board, be that permanent perennials in lower productive areas of the farm, also a good idea. But certainly if we could get some kind of a perennial phase in the rotation would also be fantastic. Again, variety selection. Can we choose varieties or species that have bigger roots? Can we look at some of those older, more traditional varieties? They're good candidates for this, but I'm sure even amongst modern varieties, there's going to be some that produce more roots than others. So is there some variety selection that we can make towards more root biomass? And or different species, is there certain species that you could maybe introduce into the rotation that are also renowned for being better root biomass producers? Soil cover, living roots, soil health principle, living roots 24/7, a really nice visual example of this. Here we have some maize just undersown with some ryegrass, cheap seed doesn't have to, okay, it could be a fancy cover mix there with a few other species, sure. Maybe a more diverse diet, especially if you were going to graze it. But even still, just look at the benefits in terms of soil protection of that cover. It's just a no-brainer in my mind. Visually, we can see that the benefits there. And of course, what does that mean? It's green, it's photosynthesizing through to as much as it can through to the end of the fall. It'll hold on to every little bit of warmth and temperature and sunlight that it can. And during that whole time, it's got those root exudates turned on, producing those root exudates, feeding that soil microbiology that is going to then grow that biomass and form that necromass that we talked about. Okay, so living roots, keeping the soil covered, it's a no-brainer. Again, just a nice visual of that. That clover will be there in the understory after harvest, and just a nice visual example of a cereal legume companion there. Things like relay cropping. You guys down there in the US have a little more opportunities for this, but I think that's also a great idea. The more you can relay crop, the more you're extending the amount of time that we have living roots in the soil. So all of those discussions about kind of relay intercropping, I think, is also great and a good opportunity there. And of course, okay, minimizing tillage. I talked about how carbon is stabilized in the soil, either chemically through that organo-mineral complexes or physically and trapped and embedded within the aggregates. When we cultivate the soil, it is those aggregates that are highly, highly vulnerable. They are the ones that are broken down, broken apart. And when we break apart that aggregate, just remember that little picture I showed of the little root hair that was embedded within that aggregate. As soon as we open up that aggregate, now that root hair is exposed to the air, exposed to oxygen, exposed to microbes. They can now access that carbon and they will rapidly feed and potentially oxidize some of that off. So yes, however we can minimize soil physical disturbance, the better. And if that means going no-till, excellent. If you want to do, if there's a compromise for some situations, okay, strip till could be a good example, or certainly minimizing disturbance, min-till or these keeping it as shallow as possible, maybe only at the surface. So whatever, I know there's different, we could have a huge debate about that whole conversation. It's not for today, but we can all agree to the mantra of minimizing disturbance. However your farming system can, however it fits within your farming system, everybody can take steps towards minimizing soil disturbance. And again, maybe a little bit of diversity. Again, just a picture of, again, some of those different roots we talked about. Different roots, plant species produce different root exudates. They have different root biomass, different root architectures, all sorts of things. So, there may be some benefits there towards that greater diversity of some of those various roots and root kind of interactions. And again, even again, choosing companions. I really like this idea of choosing a companion for its roots, choosing the cash crop for its shoots, for your yield, for your economic production, but choosing maybe some companions that maybe root more heavier rooters. Maybe they don't produce so much biomass above ground, which is great. If you want to keep that for the cash crop, that's great. You can have a companion that's more below ground. And okay, this is an example from Australia, where we've got, as you can see, radish and faba bean, along with our cereal. Now, the companions in this instance are sprayed out. Okay, so the radish and the legume will be sprayed out as the season gets towards the end, as it gets drier. So those two will go, and we're left with the monoculture of the cereal at the end. So, even this kind of strategy, I think, is also a great one. And I love this idea that we're choosing companions for their roots, because we've talked about how important roots are, obviously. So this, again, strikes a really nice balance between, practices that help to feed the soil and enhance soil, and practices that give me a yield, an economic return. We can't always sure, we'd all like to just feed and take care of our soil, but that sometimes can come at an economic cost. We don't want to be this way either. That's going to lead to environmental degradation. We've got to strike this kind of balance between environment and economics. And I think that's a really nice visual example there. And as we touched on, it's all about those roots, the benefits of cover crops, the benefits of your cash crops, to building soil organic matter, to soil health. It's all about those roots. So make sure you get out there and look at the roots. Don't adjust if you've got some cover crops, or don't judge their success by what's happening above the ground. You got to have a look at what's happening below ground. It's the roots that play that starring role in helping soil health. So get out that spade and have a look down below at the root system. And again, here's just a nice visual example of that. This is, again, it was a bit of a cover crop that was sown quite late in the season, didn't really have much time to produce lots of biomass above ground. Some of us might look at that and think, well, yeah, that's a bit of a disappointing cover crop. And on one hand, it is. However, what's happening below ground is kind of more important. And you can see we still have some very good rooting and root biomass production here. So I would argue that although maybe this may be a few inches on top, sure, we'd all like to see more, but I would argue that this cover crop is still bringing some benefits. I can still see some pretty good root action and activity there. And of course, lastly, things like livestock and livestock integration, however we can bring animals in, is just an obvious win, I think. So if you've got the capacity to do that, if you're an integrated system, great. If not, I think it's the idea of integrating livestock has lots of advantages. So explore that. It's a great way to use crop residues, cover crops, the manure, the impact of the animal. All of these things are good for soil health when managed properly. Of course, not if we're going to overgraze. And again, just that point, if we just reflect on that slide that I talked about, the poor old microbe, if it's given a choice between litter and residues and lignin and tough material versus those exudates, it prefers the exudates. It's easy for the microbe to feed on those exudates versus the litter, the complex lignin. And again, that's one of the benefits of livestock. If we have those two materials in the field and we let the animals in, well, they can graze on some of that unpalatable to a microbe residue. They can feed on that, put that through their digestive system and turn that inefficient food source into manure and leave that on the ground for then the microbes to more easily process. So that's one of the advantages of the livestock, is that they can take that stubble and lignin and complex material and do a much better job, more efficient job at breaking it down themselves and turning that into manure. Okay, we did pretty good on time. In summary, roots and root exudates, they are the key inputs for building soil organic matter. If you want to build soil organic matter, you've got to think below ground. It's all about what's happening below ground. But then, of course, it's all about those microbial transformations. It's the microbes feeding on that material, microbes growing that microbial biomass, producing all of their microbial wastes and residues, etc., and ultimately dying, forming that necromass. That is also a really important aspect of the stabilization process. And those materials can be stabilized, as I mentioned, either chemically attached to mineral surfaces, and therefore clay and silty soils have bigger potential to form that, or wrapped up within aggregates, and all soils can form that aggregate stabilized, physically stabilized organic matter. The carbon use efficiency was something we talked about. Exudates having a higher carbon use efficiency than that complex litter, and that means that those exudates, a greater percentage of those exudates will turn into microbial biomass and soil organic matter than litter. So practically speaking, the living roots principle is really important here. Nitrogen inputs can build or burn soil organic matter, so we just want to try and optimize that. Again, be aware of excess negative effects of excess nitrogen in the system. So be mindful of this. Use organic amendments where possible. And I think legumes are, practically speaking, the easy way to bring organic forms of nitrogen through their roots, through their root exudates, through their residues as it decays. I think legumes are a good piece of this puzzle. And there's no doubt about it. Soil organic matter dynamics are complex. We do indeed have many knowledge gaps. There's been a lot of rethinking that I mentioned, a lot of new research and rethinking into soil organic matter, and I tried to summarize that this morning. But I'll acknowledge, of course, we still have so many knowledge gaps. There are still many more things to know and to go. So watch this space. I think there's a lot of really exciting rethinking happening in the space of soil organic matter at the moment. So watch this space, and there'll be some important things to come. But I think that the key point about this kind of living roots and root exudates from a practical level is kind of the key take-home message. So. Okay, thank you very much. We got there. We do have a couple of minutes for questions if you want, Daryl, or you tell me what's next.
You bet. Thank you very much, Joel, for that. We do have a few minutes. You timed it perfectly. A couple questions for Joel. We will have question and answers at the end as well, but if you have some now that can't wait, you certainly can ask them. Yes. Dane Bisey. Takes me a while to get around, Joel. I'm not the athlete I once was.
That's okay.
We'll have to talk about that sometime.
Well, thanks a lot, Joel. And you really got me thinking. In cropping systems, a lot of times people are adding nutrients to the soil to ensure their success. And it seems to go the other way when you're looking at doing a perennial planting. A lot of times we think they're magic beans, you throw them in the soil and they're going to grow. So if you were doing a diverse perennial planting for in a cropping system or for a range planting, are there steps that you would take to ensure a greater success or have it established quicker?
Yeah, okay. Good question. So perennials, the fact that they're perennials, as we touched on, they do allocate more carbon below ground and they grow bigger root systems. And one of the benefits that comes with that, let's start with the negative. One of the costs that come with that is that they are often a bit slower to establish because they're allocating more carbon below ground early on than, say, an annual, which is quicker out of the ground, quicker to get going. So they're a bit slower to establish because they're really investing in the roots. But that becomes a benefit at the latter stages where that bigger root system is then exploring a greater and accessed a greater volume of soil and soil nutrition. So that bigger root system leads to greater nutrient use efficiencies because that bigger root system is accessing a greater volume of soil nutrients, which then it can draw from for its production. So I think, therefore, yes, I agree that I would also advise modest amounts of inputs for a perennial system. I wouldn't be going heavy. You've got to play a longer term game with perennials. So I would argue some more modest inputs to get established. I definitely wouldn't overdo it, and just acknowledge that yes, they're going to take a little bit more time, but that will come with a benefit later on due to that bigger root system. So in terms of establishment, I mean, it really depends on the context. If you had any severe nutrient deficiencies or really constraints in your soil type, yeah, sure, I wouldn't be opposed to putting some seed treatments on or a little bit of starter fertilizer or a liquid inject of certain key nutrients that maybe your soils are particularly lacking in. I would always combine those with a carbon source So I talked about that kind of this idea of organic or carbon-based or carbon-bound nutrient management is a better. I'll talk about this in the next presentation, so you'll get a really clear picture on this in the next presentation. So yeah, carbon-based kind of nutrients, and yeah, maybe things like just small amounts, seed treatments, small amounts in a liquid inject, and maybe some kind of a biological inoculant like a compost extract or something to put some broad spectrum diversity around that seed to provide some microbes for establishment as well. So I would say that you'll get a really clear answer to that question as we move into the next presentation as well.
Joel, we got one more question before we have to break off.
Hi, Joel. Hey, thanks for the great talk. That was fantastic. I think you put together a real compelling argument on the role of roots for carbon accrual in soil. I wanted to get your thoughts, though, on the role of soil acidification and what effect it might have on both the chemical stabilization of carbon in the soil, but also its potential effect on root biomass and even the soil microbial biomass.
Yes, good question. And yeah, it does have a negative effect on these things. So for sure, when we have lots of hydrogen in the system, lots of acidity, it is harder. All of that hydrogen is kind of crowding out the mineral surfaces of the soil, kind of binding to and overcrowding. And so yes, it negatively affects the ability of some of those organic carbon fractions to then form those organo-mineral bonds. So yes, we know that soil carbon sequestration potential is less under acidic conditions. So if viable, if possible, yes, looking at liming materials to maybe try and bring that pH up a little bit. And this is the big problem in many soil types in Australia. We have many very acidic soils there, and there's a lot of good evidence to support that yes, that does cap your potential for carbon sequestration because of that excess hydrogen in the system. And then, yes, the other part of your question, microbial aspects, yeah, also highly acidic soils has also, yeah, the pH has a major impact over soil microbial biomass, microbial activity, and yeah, sure, generally we see that fungi are a little more adapt to more acidic conditions, so they can be more active there than bacteria. So you'll always have still some microbes. There will always be some species who will thrive or will be okay in those conditions. So it's not to say that you're going to have no potential or no microbes. There will be some acid tolerant, loving microbes that would still thrive. But the generalized answer to your question is that yes, under quite acidic conditions, generally you are going to see less overall microbial biomass. It is a negative effect for them as well.
Okay. Well, with that, Joel will bug out, and we're going to have a break now, and you'll be back at about 11:10, 11:05 in there. And again, let's give Joel a big hand. Appreciate his talk this morning. Thank you very much.